we increase speed to 0.5 knots, continue tracking? Thank you.
The ship must get so annoyed with that. 0.1 faster, 0.1 slower. It's like all the difference in the world for us. Big ship. Can you go 0.1 of a knot faster, please? Oh, for God's sake.
I think 10 meter steps must kill them too.
Yeah.
Like, really? Really?
I have a 10-meter step.
Oh.
Aaron, can you zoom out on the high pack and just give me the long view for just a couple seconds here?
Morning.
Okay.
Hey, Miles.
You guys get to go straight. Drive straight.
All right, that's fine, Aaron. You can do what you want with it. They didn't do it yesterday, did they? Ooh.
All right, everybody.
ROV
It looks
team switching
like
out. Thanks,
actually
guys.
we are at a shift change, so next shift is coming on. Please welcome Carrie. She's the next SCF. And we will see you again from 12 to 4.
Can you zoom back in on high pack so I can see the end of this? Okay, thank you. We're almost there. So then, crash landing. Okay, Magda, we're almost at the end. Okay, thank you. And then if you go to return to contents, then you can go to the table of contents, which is all linked. I just announce over audio end of transect and read off the time, and I write it down. That's okay, because I want it on audio. And then Magda puts it in the log. Yeah, they're all on a line. So we've done 3A, 3B, so you're going to do 3C, which 3C has a dog leg in it. Good luck with that. I am. I watch it.
Hi everyone, this is Carrie, the Science Communication Fellow. We're just switching watches now and getting settled, so thank you so much for your patience. We'll be back in just a moment and introduce ourselves.
Okay, end of transect Magda at 23:04:55.
Hey, Carrie, we're still getting set up here, transitioning with the end of the transect. We will get on to talk more in a little bit.
Hello, this is Danny in the watch lead seat.
Good afternoon, Danny. This is Dan in the Herc seat.
Good afternoon, everyone. So it looks like the last watch got us all set up on our new dive and plan to run these transects. So this is a little bit different mode that we'll be in than we were if you were watching the Cordell Bank National Marine Sanctuary dives. We are following some transect lines, and we want to keep those at a consistent height off the bottom, a consistent speed, so we're getting good visuals to do some quantitative analysis on these. So we just finished a transect, and we are headed from 3B to 3C, and we are off effort at this point, means we can kind of wander a little bit as long as we're not slowing the ship down. And once we get to 3C, we will start another on-effort transect.
Oh, did we hit the end?
Yes, we had end of transect at 3B.
Okay. Oh, you know the one thing I didn't ask Jess?
What?
How do I stop this thing?
Oh.
The computer is flying
Turn
that.
off
No,
auto.
don't press that button.
You can turn off
I
auto
know.
XY. How would I know? You're scaring me.
So I got the lesson on the auto XY, and that's the computer flying that razor straight line.
Great. Well, the visuals have been looking really good, so that seems to be working out.
So do you want to stop and look, or just keep on?
We're just going to
Keep
keep
it on.
moving, and you can, if you want to turn and look at anything, you can, but we don't want to slow the ship down. We just want to get to the next point.
And as we cruise, it'd be great if everyone can introduce themselves. So thanks, Danny and Dan, for doing so. Can everyone else in the room say hi, who they are, what their role is on our watch today?
This is Jenny Stock. I'm the Education and Outreach Coordinator for Cordell Bank National Marine Sanctuary, and I'm Psi 1 here assisting Danny and helping out answer questions and just loving all these cool soft bottom creatures in the sanctuary. It's really fun to get back on the seafloor this afternoon.
And I'm Taylor Ann. I am the Ocean Sciences intern and the data logger on this watch. I'm having a great time here at the EV Nautilus, and I'm really excited to see this marine sanctuary.
Uh-oh, I see some marine debris.
Cool. How about on the front row?
I'm Antonella Wilby. I'm the Argus pilot for this watch.
Still Dan, still sitting in the Herc seat, doing nothing. Pushing buttons.
My name's Wayne. I'll be the lead video engineer for this next 4 hours.
And all the way at the end, Navigator, mind introducing yourself so we can check your sound?
Roger, I'm Miles. I'm navigator for this watch. I was apparently speaking to the bridge.
Hi, Bridge.
Thanks all.
I don't know if everybody can see the camera Argus looking down, but I think I'm going to nickname this area Siphonophore City. We've been sitting in these gelatinous Siphonophores for a while. It's pretty cool.
That is cool.
I've never seen so many.
It's so awesome. They're beautiful.
Turn your lights off.
Roger.
We have some pretty enthused audience members from Monterey Bay National Marine Sanctuary Exploration Center. Hi, everyone. Thanks for tuning in.
Hi, sanctuary friends.
EV Nautilus will be heading down to you guys next week.
Ooh. Wow, that's a cool view. With the lights off, you can see the density of those Siphonophores. I just had a little coffee. I'm a little chatty. You could just tell me to be quiet.
So they're not bioluminescent? The LEDs that we see are reflections of our light?
Yes.
That's right.
Mm. I think there are some bioluminescent Siphonophores. Maybe just not here. But you'd have to turn off all the lights off Herc, too, to really see that. And we have all these lights on the monitors, too, so. So one thing I've noticed that's different on this dive so far is we've seen a lot of these Venus flytrap anemones, that common name we give it for these anemones that have these awesome tentacles sticking out, like a flytrap. And there's these ones that are kind of a deep purple here that we didn't see so much in the Cordell Box Canyon. And it's the same genus, Actinoscyphia. I'm not sure I'm saying that properly, but the orange ones are Aurelia, and we don't have a species on the purple ones, but they are closely related.
Looks like you're getting a little ahead of me.
Oh, I have to press more buttons. Serious?
I don't know.
Ship.
Well, I haven't moved the camera.
I'll slow down, see if I can do this right. Decimal 1.5. Alexa, slow down to 0.15 meters per second. Hey, it worked.
So Jenny, can you remind us where we are and why this marine sanctuary is of such great interest to the EV Nautilus in the world?
Sure. We are in an area south of Point Arena, right off the continental shelf on the slope. And we're not in a traditional canyon. I was quizzing Guy earlier, asking a little bit more about what makes a canyon. But don't ask me that question because I'm still not ready to answer it. But a deep area that we're approaching some hard substrate. And we are in the Greater Farallon Sanctuary. This is an area that was added to what was the Gulf of the Farallons National Marine Sanctuary in 2015. And the communities on land came to sanctuaries in NOAA saying, we really want national marine sanctuaries to expand up here, meaning up in the northern Sonoma coast. And through a public process and environmental impact statement and public comment periods, the sanctuary is officially expanded in 2015. And so this area was added to the Greater Farallons National Marine Sanctuary in 2015. And so we're learning more about these deep habitats offshore. Some of these areas that we are studying are being looked at by the Pacific Fisheries Management Council, which is the council that manages fisheries for the United States. They are the governing body for all the fisheries. And we are looking at areas that are considered essential fish habitat. Areas to be considered as essential fish habitat and areas that may be removed as essential fish habitat. And what that designation means is that essential fish habitat are areas that are really important to the reproduction of fishes and successful rearing of them to be able to live out their lives and be abundant. So we're really looking at some of the habitats that are in and out of these areas to better understand them so we can provide our information to that body so they can make the best decisions for the best health of the seafloor here in the sanctuary. So that's part of our mission with these transects and our Greater Farallons dives.
So helpful to have that context. Thanks, Jenny.
Sure, and if Jan wants to chime in, if she's in the lounge, she's welcome to chime in too. Of course, we're also getting to just know this habitat since we haven't been able to get this deep here before. We're also characterizing, getting to know what species we have here and hoping to find some cool things like these cool sea pens.
Hey Jenny, I heard my name, but I really wasn't paying attention. This is Jan.
I was just saying if you wanted to chime in and add anything to the purpose of these transects in terms of consideration for providing information to the Fisheries Management Council about the best use of these areas.
Yeah.
You're welcome to add to it.
So this particular area was selected because in this region, the substrate modeling does indicate, and again, this is modeling, kind of steep slope, hard rocky features. Well, we don't have steep slope, hard rocky features that we're looking at because it is a model, and models aren't always, actual true reality. So, but that's why this particular area where we landed and that we're transiting across is what was selected. The other features that we're going to be going to in several hours are currently the Point Arena south essential fish habitat conservation area is close to bottom trawling, and the Pacific Fisheries Management Council, which is a stakeholders group of agencies as well as fishing and conservation, as well as management scientists and researchers. It's basically the full suite of stakeholders decided to open up certain areas of the Point Arena South essential fish habitat to bottom trawling. And so what we are doing on this particular cruise later on today and tomorrow, we'll be looking at the Point Arena area and just seeing how confident we are with opening up these areas. And I say we as in NOAA and the Pacific Fisheries Management Council. The sanctuaries don't manage fisheries, and but we do advise our sister NOAA agencies on sensitive habitats, so those rocky features. This is a nice area for bioturbation, meaning that there's a lot of critters living in the soft sediment. There's also a lot of really beautiful tall sea pens, so we might consider some of this area after we do the analyses. But the sanctuaries in general are advisory to National Marine Fisheries Service on health and condition and ecology of our habitats.
Thank you, Jan. So that's one of the roles that National Marine Sanctuaries play, is to best advise the Fisheries Management Council and the NOAA representative to that council, National Marine Fisheries Service, with the best information we have about the habitats in National Marine Sanctuaries. The Pacific Fisheries Management Council was set up as part of the Magnuson Fisheries Act, and that act set up regional fishery management councils around the United States to look at fisheries from a larger regional perspective. And so having one council to look at fisheries from the larger ecosystem regional perspective versus tiny little areas makes more sense ecologically, economically, and we help support the best decision making with the research that we're able to do on cruises like this.
This is Danny, and I can talk a little bit about the animals that we're seeing. The mud habitat sometimes gets a bad rap for deep sea research. People don't think it's very interesting, but it's actually a really important habitat for all these species that live here, and there's actually a lot of animals that live in this habitat that we can't see because they're actually in the mud. So we're seeing a lot of these tall sea pens, which are likely halipterous sea pens, a lot of anemones. I've seen some eel pouts and grenadier fish. Sea stars. Not sure what kind that is.
I'm sure the Newcastle University Marine Biology Lab is loving this. They're all listening and tuning in right now, so thanks everyone
That's
for
awesome.
Yeah.
Welcome.
When I took marine biology, there was no telepresence going on. This is really awesome for schools all around the world to be able to watch the ocean exploration happening live.
On most famous and beautiful exploration vessel, Nautilus.
I can't argue with that. I agree.
Sea cucumbers and shrimp or prawn.
I didn't realize how many different types of sea pens there were.
There are quite a bit, and we have on board Dr. Gary Williams from the California Academy of Sciences, and he is an expert in sea pen taxonomy. So he studies a lot of all gorgonians, and specifically has some expertise in sea pens.
It's a soft coral?
It's a gorgonian coral.
Gorgonian coral.
And what does that mean? Gregorian?
Gorgonian, it's a group of corals. They are often found a lot in the deep sea, and I think that refers to a protein that those corals have, gorgonin protein. I'm not sure why they're named that. I think that might be why. Okay, we're coming up to point 3C. The ship is over 3C right now, so once the ROV gets there, I will call start of transect, and we'll get that time. I'll write that down, and we'll get that in the data logger, the data log, and then we will begin that transect.
Do we know what the intended catch would be for the commercial fishermen that are requesting bottom fishing here?
In this area, it could be for flatfish or some rockfish. They generally call that ground fish, so the ground fish fishery.
Thank you.
So I think we are not in the area that they are proposed to open yet. We are still west of that area, I believe.
You can see a little bit of rock there. It's loaded with anemones and brachiopods.
What are the brachiopods?
They're like a mollusk. They are a mollusk, and they kind of look like a scallop, but they're a little rounder. I saw some on our other dives in Bucks Canyon too, and actually we pulled up some in one of the sponge samples. There were a couple in there. They are an old mollusk that have been around a long time.
They live.
I should know more about brachiopods. Yeah, I think they filter for plankton and detritus. Live on the rocks. Let's see if I can learn more about brachiopods.
So I should know this, but what's the difference between a ctenophore and a siphonophore?
Well, ctenophores are individuals. You see one individual and a siphonophore, was it?
Yeah.
Are chains of individuals all linked up. And usually you see a long chain and it looks like a bunch of individuals kind of all attached to each other.
Do you know why ctenophores sleep so well?
Tell us.
Is this a dad joke?
Why?
Because the sea is silent.
That's good.
Total dad joke.
No. No, it's not good.
Welcome back, Wayne.
Don't encourage him.
Hi, Maria Brown, the superintendent of the Greater Farallones National Marine Sanctuary. We're so glad you're with us.
Hi, Maria.
Hi, Maria.
Maria is in charge of managing this area, so it's great to have you watching.
These ctenophores kind of look like ghosts in front of the camera, making it a little blurry. And we're also seeing basket stars that have climbed up on top of these sea pens, and they're using that as habitat to gain advantage for getting food that's moving through the water column.
I've been, these that we're seeing are serpent stars.
Oh, thank you. Serpent stars. So the basket stars are the brittle stars are the ones that kind of
They're
have
very
shorter legs.
Yeah, they're similar. They're a little smaller. They still have that central disc.
Serpent stars.
And can you remind us, why do they climb up on top of these sea pens?
They get an advantage to get the food that's moving through. So they kind of prop themselves up off the off the seafloor to get food.
Okay, we are at 3C start. So that is a mark. 2328, actually 232900.
Don't think that changed your XY.
Changing the heading, no.
Yeah, but I think your XY remains. So you'll sort of crab along until the next input.
So I have to hit that again? Is that what you're saying?
I think so, yeah.
So beautiful heading correction. Wish I could fly that
Is
well.
that the flat nose?
That is a grenadier.
So what's the other one with that real tall, spiky dorsal fin?
That is the flat nose.
Ah, okay. I'm gonna
I haven't
flip in
seen
my head.
those in a little while, but the next time I see one, I'll point it out.
Snap.
So all of this great footage, this high definition footage, is coming from Hercules. It's one of our 2 ROVs. ROV means remote operated vehicle. And it's flying due to the awesome work of our pilot in the control room with us. But Hercules, although he's the workhorse of our EV Nautilus program, he is tethered to Argus, and Argus plays a super important role as well. So Argus sits above Hercules and takes on some of that energy to allow Hercules to sample and fly and take some of this beautiful footage. And then both ROVs are tethered to the EV Nautilus, the vessel that we are on right now. And a really special part of the EV Nautilus is our telepresence capacity. On ship, we have a satellite that allows us to send in real time this video footage and data and invite participation from scientists and the community all over the world. So we're super excited to have so many people tuning in right now. I'm seeing people from the Philippines and Sweden and Brazil and Germany, Australia, New Zealand, the UK, Italy.
Oh, cool.
Yeah. Canada and the United States. Thanks, everyone.
It's really awesome to be able to look at the seafloor here. I've spent a lot of time on the coastline just east of here. In fact, if you had such great weather here, you can see the coastline. And it's so fun after spending so much time on the coast up there to be offshore and seeing this soft sediment and all these amazing critters that are using this as habitat and enjoying the healthy food abundance that's in the Greater Farallones National Marine Sanctuary.
One question from a viewer is, what are those depressions in the sediment from? Do we know what animal?
I don't know what those are, but there's a few things that could be. There's a lot of animals that live in the sediment, so it could be different kinds of worms or bivalves. I'm seeing kind of like a lot of worm tubes or casings on the seafloor, so it could be from that. And sometimes you'll hear that referred to as bioturbation, because it's been the biology that has kind of been stirring that up.
And some of that bioturbation might be food for other animals. So important.
And Danny, can you tell us a little bit about the anticipated sampling that we may do on this dive?
Right, we're focused a little less on sampling for this dive, but we will be doing some sediment cores and some water sampling. The water sampling that we're doing, we're looking at both eDNA, so cells in the water that are from animals, and looking and filtering that out and telling what animals are in the water column just based on the DNA that we filtered out from the water. And then the carbonate chemistry we can also get from those same water samples. And then we're looking at sediment cores. Both the water chemistry and the sediment cores are we're working with scientists at Bodega Marine Lab who will be analyzing those. And they are looking at some of the organisms that live in the sediment, especially ones that may have settled out into the sediment a long time ago and fossilized. So you can look at stratification of those animals that historically would live in this area. We'll be doing less sampling of biological specimens just because we are trying to focus on these visual surveys and cover ground that way.
Relative to Cordell Bank, has the Greater Farallones been surveyed less comprehensively?
Well, Greater Farallones has a much larger habitat, has much larger area, I'm sorry, to survey, and this area that we're in right now has only been part of the sanctuary since 2015, so they've had less time to characterize it. And Greater Farallones has made a lot of effort to characterize their area, I would say, I think since 2010 maybe, and 2012 were some of the early surveys looking at the habitat with an ROV down closer to San Francisco in the original sanctuary area. They have some features down there, some underwater features like the Farallon Escarpment and some other rocky habitat areas that they've looked at, kind of similar to Cordell Bank. So Cordell Bank has had has focused on the bank itself, was a real focus of the sanctuary before 2015 and has been surveyed for decades and with using different technology, and Greater Farallones has made great strides in surveying their sanctuary more recently.
This area that we're in was just mapped by the Nautilus in May. So the Nautilus was out here mapping it to help inform how to do these surveys. So mapping is a key part of planning surveys to have good resolution data to best target where these deep ROV operations would best be conducted. So it's great to have that data that Nautilus was able to get in May, and Sage was part of that, coming out here, helping to get that data. We're lucky to have Guy Cochran on board with us too from U.S. Geological Survey, who's been helping interpret mapping data for the sanctuaries for a while so we can best target our survey areas and understand what's happening based on the habitat types. These are cool sea pens.
So can I submit a viewer question?
Please.
Why are there so many ctenophores, and how old are they? Like, why are they all together like that?
Well, there are a lot of different types of jellies that we have off our coast year-round. Definitely more so in the spring and summer and fall months. And then as we get winter storms and colder water in the winter, they tend to back off a little bit because they really survive well by having a lot of plankton to eat. And you know, one thing I'm interested in is why are there so many here? We didn't see them so much down in Bucks Canyon, but I'm pondering the fact that there's been some temperatures warming. We're at the end of upwelling season, and we're into some of that transition where we go into the fall a little bit, and so the water tends to be a little warmer anyway because upwelling has slowed a little bit. Our upwelling times are really peak in the spring and the summer where we get a lot of wind and a lot of upwelling of cold water and nutrients, and the fall we tend to see as a little bit warmer period because upwelling has subsided, and so there's a lot of plankton, but that's just one guess. I have a feeling there's a lot more to the answer of this density.
I do know that the pelagic and midwater species, the gelatinous species, they're really influenced by the conditions and the currents, and so it can be very changeable up in one place from time to time. So you could come back, and tomorrow if we dove the same site, we might see different densities or different species in the water. So that's one thing that's interesting about studying those types of species.
There's some rockfish that love this gelatinous zooplankton in the midwater area. Oh, that looks like a ctenophore eating a ctenophore. I've seen that before. Burrowies eating other ctenophores. I think it's blue rockfish. They're a little bit shallower. They like to eat these jellies, ctenophores. And so what you're seeing, that kind of that radiating light, those are their little combs. There's little combs that help move food to their gut. And when the light of the ROV hits them, it shimmers. It's kind of like a disco under the ocean. Another Halloween costume to be made.
What's that? One's hit bubblegum? Oh yeah, there's tons of them down low now.
Okay, we're approaching the end of transect 3C, so when we get there, I will call end of transect.
Ooh, you got a skate up here.
There's some marine debris there. That's what I echo. Oh, it's a skate, sorry. First one I've seen on this trip. I thought it was a paper bag. That's the Bobby Argus moment there. No.
One of our viewers is curious if Siphonophores can get stuck in the ROVs and or clog or harm visibility to our cameras in any way.
If they, sorry, I heard the first part, if they get stuck in the ROV, but what was the second part?
Yeah, just if the Siphonophores can impact the ROVs in any way.
If they can impact the ROV?
Correct.
Okay, and that is end of transect at 23:43:49.
Oh. Roger. 07. I'm going to try it this way. As far as I know, they haven't been an issue. Biology in general, the biggest problem we've had is, it doesn't let me, is fish get stuck in there, and they've actually had big enough ones to get stuck and stop the thruster, which severely impacts operation. Maybe it's because I have auto head on it still. It's not letting me come around. They're just really slow. Sorry, you said 07076, Roger.
Yeah, these particular jellies are of small enough size that they will, if they get too close to the intake of any one of the thrusters,
So
they will process through the impellers and probably come out in more pieces than they went in.
And Jenny, earlier you referenced the temperature, but what other sensors does Herc have, and what else are we monitoring when we go down to these depths?
Let's see. we have salinity, temperature, oxygen concentration, that's what I see, but maybe the ROV team could fill in if there's other things that we're taking data on.
Yeah, I'll give that one a go, just because I happen to be looking at the sensors page. So we have a CTD, it's, salinity, temperature, and we talked about that one. Depth. We have an octans gyro, which provides our pitch and roll information and somewhat heading. We have 2 compasses on board the ROV, and we have, which give us, that feeds heading information to the gyro. We have a parascientific depth sensor, oxygen sensor, temperature probe, the Doppler velocity log, which tracks the seabed and also provides us with altitude information. And Argus also 2 compasses and a parascientific depth sensor and an altimeter. Sub-bottom profiler, 2 sonars. Should I keep going?
I think that covers it.
Yeah, that was great.
there's a few more, but
Comprehensive.
I see your eyes are rolling into the back of your head. So.
For those that were watching on the quad view, we did have one of the Siphonophores that was hung up on the bubble cam for a moment, and it occluded our view for a bit. But he fell off.
See if I can make it go from scratch. Is that a good heading still, Miles? Or? 1, 2, 3. 7.5. What's that?
You can change the goal exactly. I think. No? You can change the step.
Got it.
It's weird you can't change it.
And one of our returning viewers was also curious about the lasers and just wanted a reminder on the distance between them, and if you guys could just tell us though the purpose.
10 centimeters or about 4 inches, whichever comes first.
And are they only used for the to understand measurement, or does it help in any way with cameras?
Yeah, they give scaling obviously to what they're hitting, but it helps us tremendously. What happened there? Sorry. It gives a reference when the scientists are asking the pilots to look at something in particular, and then we're going to zoom in on it. So we say above the lasers or below the lasers, or to the right or left.
It's giving XY.
The lasers, since they're parallel, anything it hits, we know that the distance between those 2 green dots is going to be 10 centimeters or 4 inches,
Miles,
whichever
you
comes
could just
first.
call him nav. That's what I do.
So we can tell if we have a tiny fish that's very close to the lens or a large fish that is further away from the lens. They're both going to take up the same number of pixels on the screen, but depending on its distance from us will determine, you know, if we can determine its actual true length. So those lasers would give us the ability to calculate the size of organisms that are in frame.
What's that?
I have some cool Siphonophore trivia for you.
Oh boy.
Because
why would you?
they're just so cool, and I have an internet connection here, so I wanted to learn more. Siphonophores are multicellular, few tissues, some organs and organelles. They have an internal cavity and a mouth with anal pores. The cilia we're seeing, so that those combs are actually cilia, and that's how they swim. So they're moving cilia to swim. They are mostly...
Okay, pilot and Nav, we are going to hold here while we sort out our next move, because we have completed some of the transects that we wanted to do, so it looks like we can alter the plan a little bit and maybe visit some different habitat.
Herc copies.
We'll talk about Siphonophores after we get resituated here.
We'll see how close we can get to a Siphonophore.